Welcome back to the deep dive. Our mission today is really laser-focused, designed specifically for you, the learner, especially if you're navigating first-year medicine.
Right.
We are tackling, well, one of the most conceptually rich topics, the biochemistry of vitamins.
Absolutely fundamental.
Think of this as your essential guide, maybe fast track, to understanding not just the names, but the underlying molecular mechanisms, the things that link a single deficiency to sometimes a really dramatic clinical outcome.
Indeed. We're zooming right in on these tiny organic compounds, these essential nutrients. They're absolutely critical for, well, everything: growth, health, welfare.
The unsung heroes.
Pretty much. They're the unsung heroes of metabolic pathways, usually acting as coenzymes. If you think of the body's machinery as a factory, vitamins are those crucial, non-replaceable tools that keep the production lines running.
Yeah, and we know this section can sometimes feel like just a massive list of facts to memorize.
It can feel that way.
But our goal here is to give you a rapid, strong conceptual framework. We want to move past that rote memorization and show you the clinical why. Why does this molecule matter? We want you to grasp that, say, a swollen tongue or sudden memory loss is actually just the downstream result of a specific blocked enzymatic pathway.
And the big picture here really is evolution. We, as higher organisms, somewhere along the line, lost the ability to synthesize these vital molecules.
Which things like bacteria can still do.
Precisely, like E. coli. Now, because our body degrades them relatively slowly, we only need them in tiny amounts, but, and this is the key, they must come from the diet.
Okay.
So by the end of this deep dive, you should be able to connect those deficiency symptoms directly back to a broken coenzyme function. And that's really the definition of clinically relevant biochemistry.
Okay, let's unpack the history then. How were these things actually found and named? It's a kind of a fascinating story, isn't it? Gradual realization starting back in the early 20th century.
It really is.
Before vitamins were even called vitamins, they had another name: accessory factors.
That's it. Credit for that goes to Hopkins. He coined that term because he realized that animals fed only purified carbs, fats, proteins. They just failed to thrive.
Something was missing.
Exactly. He hypothesized there must be some unknown essential nutrients in natural foods. That was really the intellectual spark. Then, jump forward to 1913, a Polish chemist, Casimir Funk.
Ah, Funk, I remember that name.
Yes, he isolated an active principle from rice polishings, the stuff that could cure beriberi.
And that's where the name came from, right? Even though he was a bit off target about the whole group.
Precisely. Because the specific compound he found was an amine. He called it vitamin, literally life amine.
Okay.
Now, he was incorrect that all of them were amines. Vitamin C proved that later that the name stuck. They just dropped the final E eventually.
So we go from accessory factors to vitamin, and then we plunge into this, well, slightly confusing alphabet soup. How did we actually get A, B, C, and then jump to K? What happened there?
Right. Well, the foundational alphabet system really came about in 1915, McCollum and Davis. They classified them based on solubility.
Simple enough to start.
Yeah, just two initially. Fat-soluble A and water-soluble B. But then research really took off. Vitamin C, the anti-scurvy factor, was identified pretty quickly.
Okay.
Then researchers realized that what they call vitamin A was actually two factors. There was the anti-night blindness factor, which kept the name A, and the anti-ricket factor, which became vitamin D.
And the naming just got very practical after that, reflecting function more than chemistry.
Absolutely. We got vitamin E, known then as the anti-sterility factor.
Yeah.
And then, vitamin K is interesting. They skipped F.
Why?
Named it K for coagulation. That's coagulation in German, directly pointing to its role in blood clotting. Very functional naming.
And the B complex group. That's where the alphabet system really went wild. B2, B3, B12, and then a bunch of numbers just vanished.
Yeah, that was a complex mixture. It took a lot of work in the 1920s and 30s to sort it out. As they got better at purifying things, scientists realized many of those numbered B vitamins like B4, B8, B10 were actually just mixtures of vitamins they already knew.
I see.
Or they weren't really essential nutrients for humans after all. So today, we still use the numbers for B1, B2, B6, B12, but for the others, we mostly use their chemical names: niacin, folic acid, pantothenic acid. It shows a more mature understanding, you know?
Right. Which brings us neatly to the definitive classification, the one that really matters clinically: solubility.
That is the critical takeaway. It
Yeah.
dictates so much about how they behave. So the 15 essential human vitamins are split.
Okay.
Fat-soluble,
Yeah.
A, D, E, and K,
Got it.
and water-soluble. Vitamin C stands alone as non-B complex,
Yeah.
and then you have the whole B group, the B complex vitamins.
And within that B complex group, there's that really useful clinical subdivision. It connects the vitamins straight to its main job in the body.
Yes, we group them thematically. It helps make sense of them. You've got the energy-releasing group, B1, B2, B3, B5, B6, B7.
Why energy releasing?
Because their coenzymes are core components of, catabolic pathways. Think TCA cycle, breaking things down for energy.
Makes sense.
And then you have the hematopoietic group,
Mm-hmm.
folic acid, which is B9 and B12.
Hematopoietic meaning blood cell formation.
Exactly. They're absolutely necessary for cell division, particularly making red blood cells. It's also important to note most water-soluble vitamins form coenzymes, but among the fat-soluble ones, only vitamin K has a really clearly defined coenzyme role.
Now, here's a point. You mentioned synthesis by intestinal bacteria. Let's pause on that for a second. If our gut bacteria make some vitamins, why do we still need to eat them? Seems like a bit of a contradiction.
It does seem that way, but by definition, vitamins are things we can't synthesize sufficiently. Now, gut bacteria, mostly living down the colon where absorption isn't great for many things, they can produce significant amounts of certain vitamins. It's generally accepted that normal gut synthesis of vitamin K and biotin, B7, might actually be enough to meet our body's needs, under normal circumstances.
Okay, K and biotin potentially covered by gut bugs. What about the others?
For the other B-complex vitamins, the contribution is probably less reliable, generally not enough on its own to prevent deficiency if your diet is poor.
So this sounds like a biochemical loophole, but it leads to a really crucial clinical point. If you have a patient on strong, broad-spectrum antibiotics, what should you immediately think about?
You absolutely need to think about supplementation. Those antibiotics often wipe out the beneficial vitamin-synthesizing bacteria along with the pathogens.
Like collateral damage.
Exactly. This can lead to a secondary deficiency, particularly of vitamin K, which can affect clotting and maybe some B-complex vitamins. So yes, recommending extra intake or supplements during and sometimes after antibiotic therapy is often a very good idea.
Okay, let's dig into what solubility actually means for these vitamins. How does it affect storage, transport, and importantly for you as future clinicians, toxicity? Let's start with the fat-soluble group, A, D, E, and K.
Right. Their fundamental properties all stem from being lipophilic fat-loving. They dissolve in fats, oils, and fat solvents like alcohol or acetone.
And that affects how we absorb them.
Absolutely. Absorption requires dietary fat. You need bile salts to emulsify things, form cells, the whole fat absorption pathway. And then they're transported bundled up in chylomicrons and lipoproteins in the blood.
And because they like fat, they don't just wash out easily, do they?
Exactly. That's the key difference. They get stored, often in large amounts, mainly in the liver and adipose tissue. They're not readily excreted in urine.
Okay, stored. That sounds good, like a backup supply.
It is, but it's a double-edged sword. It means we don't necessarily need a constant daily supply, which is convenient, but it also creates a major clinical risk.
And here's where it gets really interesting and maybe a bit scary for medical students. The danger of overdose. Hypervitaminosis.
Yes. Because they are stored and stick around, taking in too much, especially vitamin A and vitamin D, leads to chronic accumulation. This buildup causes significant toxic effects.
Are E and K also toxic?
Much, much less so. Vitamin E and K are far less toxic in excess. But the threat of hypervitaminosis A and D, that's a primary clinical concern. It's something you almost never see with water-soluble vitamins.
Interesting. And chemically, you mentioned they have a common structure.
Yeah, it's a neat chemical note. All the fat-soluble vitamins, except vitamin K, are structurally isoprenoid compounds. They're built from repeating 5 carbon units, these isoprene units. Vitamin K is the odd one out structurally, too.
Okay, let's switch gears. Water-soluble vitamins, that's C and the B group. What's the defining characteristic here regarding storage?
Well, chemically, they're a mixed bag, quite heterogeneous. Their only real commonality is that they dissolve in water.
And that means?
It means they're readily excreted in the urine. They get flushed out easily. So generally, they're non-toxic even in high amounts. And with one significant exception, they are not stored in large quantities.
Okay, what's the exception? And why does that matter for, say, how quickly a deficiency shows up?
The big exception is vitamin B12, cobalamin. It's unique. The liver can actually store quite a bit of it, enough to last for several years sometimes.
Wow, years.
Yeah, quite a while. But for all the other water-soluble vitamins, the body maintains negligible reserves. Basically, what you take in today is used or excreted pretty quickly.
So you need a continuous supply.
You absolutely do.
Yeah.
Continuous dietary supply is necessary. If intake stops, deficiency symptoms can appear relatively fast, often within weeks or maybe a few months, depending on the vitamin.
And this rapid turnover, this lack of storage, it also helps explain why deficiencies often show up together, right? It's rarely just one thing missing.
That's a very important clinical point.
Yeah.
Yes, if a patient has a poor diet, it's seldom just one water-soluble vitamin that's low. Deficiencies are often multiple and overlapping. This can make the exact clinical diagnosis tricky sometimes. Now, the B-complex vitamins, function-wise, mostly form coenzymes involved in either energy generation or hematopoiesis, making blood cells. When these pathways get impaired due to deficiency, you often see a sort of common constellation of symptoms.
What kind of symptoms are we talking about?
Things like dermatitis, skin issues, glossitis, that's a red swollen tongue, sometimes described as magenta or purplish. Cheilitis, those painful cracks at the corners of the lips. Diarrhea can happen. General malaise, feeling unwell, and sometimes mental confusion.
But if we look closer, you mentioned some B vitamins have a particularly strong link to the nervous system, more so than others.
Absolutely. While several deficiencies can have neurological effects, deficiencies of B1, thiamine, B6, pyridoxine, and B12, cobalamin, are most closely and specifically associated with severe neurological problems.
Why those three in particular?
It often comes down to their specific roles, maybe in making crucial neurotransmitters or in maintaining the integrity of the myelin sheath that protects nerves. We'll get into the specifics for each one.
All right, let's dive into the individuals, starting with the fat-soluble ones. Vitamin A, the retinoids. This one's kind of famous, right? Even ancient remedies involved it, like Hippocrates using ox liver for night blindness.
That's right, a very old observation.
Okay.
The term retinoids is broad. It covers all the natural and synthetic forms that have vitamin A activity. But the key natural players, the vitamins you need to know, are retinol.
Which is the alcohol form.
Yes, the alcohol form. That's the main one circulating and stored, usually as an ester, retinol ester. Then there's retinol, the aldehyde form. Retinol and retinol can be interconverted.
Okay.
And then there's retinoic acid, the acid form. Now, here's a crucial biochemical detail. Once retinol is oxidized to retinoic acid, that reaction is irreversible. It cannot be converted back to retinol or retinol.
Why does that irreversibility matter?
It matters because retinoic acid, while very active in regulating genes, cannot function in the visual cycle. Only retinol can do that. So the acid form supports growth and differentiation, but not vision.
Got it. And what about the plant source, beta-carotene?
Ah, yes. The provitamin A beta-carotene is what gives carrots their color. It has to be cleaved in the intestine enzymatically to yield 2 molecules of retinol.
So plants give us the precursor.
Exactly. But clinically, you need to remember, this conversion isn't super efficient. Beta-carotene only has about 1/6 the vitamin A activity of preformed retinol from animal sources. Important when you're assessing dietary intake.
Okay, let's get into its most famous function, vision. Wald's visual cycle. How does a chemical flip in retinol actually let us see in dim light?
It's a beautiful mechanism, really happening in the rod cells of the retina. The key molecule is the photosensitive pigment, rhodopsin.
What's rhodopsin made of?
It's a protein opsin linked covalently to one specific isomer of retinol, 11-cis-retinol. Now, when a photon of light hits that rhodopsin molecule, bang. The primary event is the light energy causes the 11-cis-retinol to instantly isomerize, to flip into the all-trans retinol form.
So light changes the shape of the retinal part.
Precisely. And this shape change in retinol forces a dramatic conformational change in the opsin protein it's attached to. This activated complex is called rhodopsin 2.
Okay, the protein changes shape, but how does that become an electrical signal that goes to the brain? That seems like a big leap.
It happens through a signaling cascade, the cGMP visual cascade. Rhodopsin 2, the activated form, bumps into a G protein called transducin.
Like other G protein systems.
Exactly like them. It causes transducin to swap its bound GDP for a GTP, activating it. Activated transducin then turns on another enzyme, cGMP phosphodiesterase.
Which does what?
It rapidly breaks down cyclic GMP, cGMP. Now, normally, in the dark, high levels of cGMP keep certain sodium channels in the rod cell membrane open.
So when cGMP levels plummet because the phosphodiesterase got activated,
The sodium channels slam shut. This stops the influx of positive charge, and the rod cell membrane actually becomes hyperpolarized, more negative inside.
and that hyperpolarization is the signal.
That hyperpolarization is the excitatory neural signal that gets transmitted through the optic nerve to the brain. Its counterintuitive closing channels causes excitation here, and the clinical link. In vitamin A deficiency, you don't regenerate rhodopsin efficiently. So the dark adaptation time, the time it takes your eyes to adjust to dim light, gets significantly longer. That's night blindness.
Fascinating. Okay, beyond vision, vitamin A acts more like a hormone, doesn't it? Affecting genes.
Yes, absolutely. Retinol and especially retinoic acid are critical regulators of protein synthesis via gene expression. They bind to specific nuclear receptors, much like steroid hormones do. This binding alters the transcription of hundreds of genes involved in cell growth, differentiation, and maintaining healthy epithelial tissues. It's crucial for keeping tissues like skin, lungs, and the gut lining healthy and moist. A key role is preventing inappropriate keratin synthesis in places where it shouldn't happen, like the cornea.
And that structural role, preventing keratinization, leads directly to the other major deficiency signs beyond night blindness.
Exactly. So the earliest symptom is night blindness, nyctalopia. But if the deficiency continues, you get xerophthalmia. That literally means dry eye.
Why dry?
Because the conjunctiva and cornea start to undergo keratinization. They lose their moist epithelial lining. If you look closely at the conjunctiva, you might see these white, triangular, foamy-looking plaques. Those are called Betau spots. They're quite characteristic.
Is that reversible?
Xerophthalmia and Betau spots are generally reversible with treatment. However, if it persists, it can progress to keratomalacia. That's where the cornea itself softens, liquefies, and gets irreversibly destroyed. It leads to permanent blindness, a major cause of preventable blindness in children worldwide.
Grim. And what about the flip side? Toxicity. Hypervitaminosis A, you mentioned it's a risk.
It is a serious risk, especially with chronic high intake of preformed vitamin A supplements. Symptoms can include dermatitis, skin peeling, hair loss, severe headaches from increased intracranial pressure.
Like pseudotumor cerebri.
Exactly. And liver enlargement, even cirrhosis in severe cases.
Yeah.
The toxicity seems related to excessive levels of free retinol floating around.
How does free retinol cause damage?
High levels of free retinol can destabilize cellular membranes, particularly lysosomal membranes. This causes lysosomes to leak their powerful digestive enzymes, these lysosomal hydrolases, into the cell cytoplasm, leading to widespread cellular damage.
But wait, we know people eat tons of carrots, which are packed with beta-carotene, and they don't get vitamin A toxicity. Why is beta-carotene safer?
That's a great point. Beta-carotene itself has independent benefits as an antioxidant. It scavenges free radicals, potentially reducing risk for some cancers and heart disease, separate from its vitamin A role.
Okay.
But crucially, the body tightly regulates the conversion of beta-carotene into retinol in the intestine. Once vitamin A stores are sufficient, the enzyme that cleaves beta-carotene slows down dramatically. So you just don't absorb enough to cause vitamin A toxicity from plant sources alone. Your skin might turn a bit orange, keratonemia, but it's harmless.
Right, the body has a control mechanism for the precursor. Makes sense. Okay, moving on to vitamin D, the sunshine vitamin. You said it functions more like a hormone, specifically a steroid-like hormone involved in calcium.
Absolutely. Fundamentally, it acts as a hormone. We encounter two main forms: ergocalciferol, that's D2, mainly from fortified foods or plant sources, and cholecalciferol, D3, which we get from animal products, or, importantly, synthesized in our skin.
How does the skin synthesis work?
Our skin contains a precursor, 7-dehydrocholesterol, which is derived from cholesterol. When ultraviolet B, UVB light from sunlight hits the skin, it provides the energy to convert 7-dehydrocholesterol into vitamin D3.
So sunlight is key, but D3 itself isn't the active form, right? It needs activation.
Correct. The activation is a critical two-step hydroxylation process happening in two different organs.
Okay, step one.
Step one happens in the liver. Vitamin D3, or D2 from the diet, travels to the liver and gets hydroxylated at position 25. This forms 25-hydroxycholecalciferol, often written as 25-OHD3. This is the major storage and circulating form of vitamin D, the one usually measured in blood tests to assess status.
But still not fully active.
Still not fully active. Step 2 is the really tightly regulated rate-limiting step. It happens exclusively in the kidney.
What happens there?
The 25-OHD3 travels to the kidney, and an enzyme called 1-alpha-hydroxylase adds a second hydroxyl group, this time at position 1. This enzyme requires cytochrome P450. The final product is 1025-dihydroxycholecalciferol, written as 1025-OH2D3, also known as calcitrol. That is the fully active hormone form of vitamin D.
Okay, so the kidney step is the crucial control point. What controls that enzyme, the 1-alpha-hydroxylase? This must be where the body manages calcium.
Exactly. The activity of 1-alpha-hydroxylase is under tight hormonal control, primarily responding to calcium and phosphate levels. Low plasma calcium is the main trigger.
How does low calcium activate it?
Low calcium stimulates the parathyroid glands to release parathyroid hormone, PTH. PTH then travels to the kidney and stimulates the 1-alpha-hydroxylase enzyme. So Low calcium, high PTH, more calcitriol synthesis.
Is phosphate involved too?
Yes. Low plasma phosphate levels also directly stimulate the 1-alpha hydroxylase, independent of PTH, and high levels of calcitriol itself actually feedback inhibit the enzyme, closing the loop.
Okay, so we make active calcitriol. Where does it act, and what does it do to fix low calcium?
And calcitriol, the hormone, acts primarily at 3 main sites to raise plasma calcium and phosphate levels back to normal.
Site 1.
1. The intestine. This is arguably its most important action. Calcitriol goes to intestinal cells and, acting like a steroid hormone, increases the gene expression and synthesis of a specific calcium-binding protein, carbondin. This protein dramatically boosts the absorption of both calcium and phosphate from the food we eat.
Okay, sucks more calcium from the gut. Site 2.
2. Bone. Here its action is a bit more complex, working alongside PTH. It helps coordinate bone remodeling. It does stimulate osteoblasts to lay down new bone, which requires calcium, but importantly, it also stimulates osteoclasts to resorb bone, releasing calcium and phosphate into the blood when levels are low.
So it can take calcium out of bone too?
Yes, if needed to maintain blood calcium levels, which are critical for nerve and muscle function. Site 3.
Site 3.
3. The kidney. Calcitriol acts on the renal tubules to increase the reabsorption of calcium and phosphate, minimizing their loss in urine. So it makes sure we hang on to the minerals we have.
So intestine, bone, kidney, all working to keep blood calcium stable. What happens if this system fails due to vitamin D deficiency?
The consequences are primarily skeletal. In growing children, the failure to mineralize the newly forming bone and cartilage growth plates leads to rickets.
What does rickets look like?
The bones become soft, pliable, and weak. You see characteristic bowing of the legs, bow legs, thickening at the wrists and ankles, maybe delayed closure of the fontanelles. Lab tests classically show low or normal calcium, low phosphate, elevated alkaline phosphatase, reflecting frantic bone turnover, and of course, low 25-OHD3 in calcitriol.
And in adults, it's not rickets then?
In adults whose growth plates have closed, the deficiency causes osteomalacia. This is essentially soft bones in adults. The existing bone matrix is okay, but it gets progressively demineralized. Bones become weak, prone to fractures, and patients often experience bone pain and muscle weakness.
Okay. And you mentioned a special case related to kidney failure.
Yes, renal rickets or renal osteodystrophy. This happens in patients with chronic kidney disease. Their damaged kidneys lose the ability to perform that crucial second hydroxylation step. They can't make enough active calcitriol, even if they have plenty of 25-OHD3.
So treatment has to be different.
Exactly. Giving them regular vitamin D, D2 or D3 won't help much. They need to be treated directly with the active hormone, calcitriol, or a synthetic analog to bypass the faulty kidney activation step.
Given how powerfully vitamin D affects calcium mobilization and absorption, it makes sense you flagged it as potentially very toxic in overdose.
It is. In fact, vitamin D is generally considered the most toxic of all vitamins if taken in excessive amounts. Sometimes toxicity occurs at levels only 10 to 100 times the recommended daily allowance.
What does the toxicity look like?
Hypervitaminosis D leads to severe hypercalcemia, dangerously high blood calcium levels. This causes a cascade of problems: nausea, vomiting, constipation, dehydration, confusion. But the real danger is the metastatic calcification.
Meaning calcium deposits where it shouldn't?
Precisely. Excess calcium starts depositing in soft tissues throughout the body, particularly the kidneys, leading to kidney stones and kidney damage, blood vessels, contributing to atherosclerosis, lungs, and heart. It also causes bone demineralization as the body tries to cope. It's a serious situation.
Right. Okay, definitely want to be careful with supplements. Let's move to the third fat-soluble, vitamin E. The tocopherols, sometimes called the anti-sterility vitamin historically, or maybe a vitamin in search of a disease in humans.
Both descriptions have some historical basis. The main active form you need to know is alpha-tocopherol. Its defining function is as a lipophilic antioxidant.
Lipophilic meaning it likes fatty environments.
Exactly. Because it's fat-soluble, it integrates itself into the lipid bilayers of cell membranes, think mitochondrial membranes, ER membranes, and also into circulating lipoproteins like LDL cholesterol. It sits right there in the fatty environment.
How does that location relate to its antioxidant job?
It makes it perfectly positioned to be the first line of defense against lipid peroxidation. It acts as a crucial chain-breaking antioxidant. It intercepts and neutralizes lipid peroxyl radicals.
What are those?
Those are damaging free radicals formed when polyunsaturated fatty acids, PUFAs, in membranes get attacked by reactive oxygen species. Vitamin E sacrifices itself. It gets oxidized to a quinone form, in the process thereby stopping the chain reaction of light damage that would otherwise destroy membrane integrity.
So it protects membranes from free radical damage. What are the clinical consequences of that?
Well, it helps protect red blood cells, RBCs, from oxidative stress and hemolysis, makes them less fragile. And importantly, it helps protect LDL cholesterol from oxidation. Oxidized LDL is a key player in the development of atherosclerotic plaques, so vitamin E potentially plays a role in heart health.
You also mentioned synergy earlier. It works with another nutrient.
Yes, very important synergy with the trace element selenium. Selenium is a component of the enzyme glutathione peroxidase.
What does that do?
Glutathione peroxidase is another key antioxidant enzyme. It breaks down hydrogen peroxide and lipid hydroperoxides after they've formed. So you can think of vitamin E as preventing the fire from stopping the chain reaction, and selenium glutathione peroxidase as putting out any small fires that do start cleaning up peroxides. They work together.
Okay, if it's so important for protecting membranes, why is a clear, distinct deficiency disease so rare in humans? Why in search of a disease?
It's mainly because vitamin E is very widespread in the diet. Vegetable oils, nuts, seeds, leafy greens, and the body is pretty good at recycling it. So outright dietary deficiency is very uncommon, except in severe fat malabsorption syndromes or certain rare genetic conditions affecting its transport.
What happens if severe deficiency does occur?
Symptoms are generally mild and often nonspecific. You might see increased RBC fragility leading to a mild hemolytic anemia. There can also be some neurological symptoms like ataxia or neuropathy, thought to be related to oxidative damage to nerve cell membranes over time. But compared to A or D deficiency, it's much less dramatic.
And toxicity?
Also generally considered the least toxic of the fat-soluble vitamins. Very high doses might interfere with vitamin K function and increase bleeding risk, especially in people on anticoagulants, but it doesn't cause the kind of systemic toxicity seen with A or D.
Right. Okay, our final fat-soluble vitamin, vitamin K. The odd one out, you said, because it has a specific coenzyme function.
Yes, the only one in this group with a clearly defined role as a coenzyme. Vitamin K, named for coagulation, is all about blood clotting or hemostasis.
What are the different forms?
There are several related compounds, all naphthaquinone derivatives. K1 is phylloquinone, found in green leafy vegetables. K2 is menaquinone, which is synthesized by our own intestinal bacteria and also found in some animal products. And K3 is menadione, a synthetic form that can be converted to K2 in the body.
Okay, what is the exact coenzyme mechanism? You mentioned post-translational modification. What does that mean?
It means it modifies proteins after they've been synthesized on the ribosome. Specifically, vitamin K is required for the proper function of several key blood clotting factors. Factors 2, which is prothrombin, 7, 9, X, and X.
How does it modify them?
These factors are synthesized in the liver as inactive precursors called zymogens. For them to become active, they need a special modification. Vitamin K acts as an essential coenzyme for a liver enzyme called gamma-glutamyl carboxylase.
What does that carboxylase do?
This enzyme adds an extra carboxyl group, -COH, to specific glutamic acid, GLU residues, within those precursor clotting factor proteins. This converts GLU into a modified amino acid called gamma-carboxyglutamate, abbreviated as GLA. This reaction needs vitamin K, oxygen, and carbon dioxide.
So vitamin K helps add a carboxyl group to glutamate residues on clotting factors. Why is making this GLA residue so critical for clotting? What does it do?
The GLA residue is the key. Having two adjacent carboxyl groups, instead of just one on regular glue, gives that part of the protein a strong negative charge. This allows the GLA residues to act like claws, binding very effectively with positively charged calcium ions, Ca2+.
Okay, GLA binds calcium. Then what?
This GLACAT2+ complex forms a bridge. It allows the clotting factors to dock onto the negatively charged phospholipid membranes exposed on the surface of activated platelets at the site of injury. This docking concentrates all the necessary clotting factors right where they need to be, dramatically accelerating the cascade, especially the conversion of prothrombin, factor 2, to thrombin, which is the central enzyme of coagulation. Without GLA, the factors just float around uselessly.
Ah, that makes perfect sense. And this mechanism neatly explains how common anticoagulant drugs work, like warfarin.
Exactly. Drugs like decumarol, originally found in spoiled sweet clover, and its synthetic cousin warfarin are vitamin K antagonists. They don't block the carboxylase directly. Instead, they block an enzyme called epoxide reductase.
What does epoxide reductase do?
During the carboxylation reaction, vitamin K gets converted into an inactive epoxide form. Epoxide reductase is the enzyme that recycles this inactive epoxide back into the active reduced form of vitamin K needed for the carboxylase to work again. Warfarin blocks this recycling step.
So the vitamin K gets used once and then can't be reactivated.
Precisely. By preventing the recycling of vitamin K, warfarin effectively depletes the active form, shutting down the synthesis of functional glycontaining clotting factors 2, CAI, I, and X. This leads to anticoagulation.
Makes sense. Now, deficiency. You mentioned it's uncommon because we get it from diet and gut bacteria.
That's right. Between dietary K1 and bacterial K2 synthesis, true deficiency is rare in healthy adults. When it occurs, it's usually due to secondary causes.
Like what?
Things like severe fat malabsorption syndromes, because it's fat-soluble, needs bile salts, etc., or conditions that disrupt the gut flora, like chronic diarrheal diseases, or, importantly, long-term use of broad-spectrum antibiotics that wipe out the K2-producing bacteria. Newborn infants are also at risk because their guts aren't colonized yet, and vitamin K transport across the placenta is poor, hence the routine vitamin K shot at birth.
And the result of deficiency.
You fail to produce functional clotting factors. The most direct consequence is impaired blood coagulation. Clinically, this manifests as an increased prothrombin time, PT, on lab tests, easy bruising, and in severe cases, spontaneous bleeding or hemorrhage.
Okay, let's switch gears completely now to the water-soluble group. Remember, these are the ones generally not stored well, except B12, readily excreted, and need constant dietary supply. We'll start with the one non-B-complex vitamin in this group, vitamin C ascorbic acid.
Right, vitamin C. Chemically, it's a hexose derivative related to glucose. Its key property is being a potent, reversible reducing agent. It readily donates electrons.
And it's unique because humans can't make it.
Exactly. Humans, other primates, and guinea pigs are among the few mammals that lack the ability to synthesize vitamin C. We're missing just one crucial enzyme in the synthesis pathway from glucose.
Which enzyme is that? This is a classic exam question.
It's L-guanylactone oxidase. This enzyme catalyzes the very last step. Because we lack it, vitamin C becomes an essential dietary requirement for us.
Okay, function. Vitamin C's most famous role is in collagen synthesis. Structure, strength, how does it work at the molecular level there?
It acts as an essential coenzyme for two key enzymes, prolyl hydroxylase and lysyl hydroxylase.
What do they do?
As procollagen chains are being synthesized, these enzymes add hydroxyl groups to specific proline and lysine residues within the chains. This hydroxylation, which requires vitamin C to keep the iron atom in the enzyme active in the Fe2+ state, is absolutely critical.
Why is hydroxylation so important for collagen?
These hydroxylated prolines and lysines are necessary for forming stable hydrogen bonds between the collagen chains, allowing them to wind tightly into the strong triple helix structure. They're also sites for later cross-linking between collagen molecules. Without proper hydroxylation, you get weak, unstable collagen fibers.
And that failure of collagen leads directly to the classic deficiency disease, scurvy.
Exactly. The horrific symptoms of scurvy, spongy, bleeding gums, loose teeth, poor wound healing, fragile blood vessels causing easy bruising and hemorrhage, eventually bone pain and weakness are all direct consequences of producing defective collagen. Connective tissues basically fall apart. Scurvy is the textbook example of a structural protein failing because its modifying coenzyme is missing.
Beyond collagen, vitamin C is also a major systemic antioxidant, right?
Yes, its reducing power makes it a very effective water-soluble antioxidant. It helps protect other molecules from oxidative damage. For instance, it enhances the absorption of dietary non-heme iron by keeping it reduced in the more soluble ferrous, Fe2+ state in the gut.
Any other antioxidant roles?
It can help regenerate the active form of vitamin E after it's been oxidized. It plays a role in reducing harmful methemoglobin back to functional hemoglobin, and generally it helps spare other vitamins from oxidative destruction.
Okay. Now, there's always been some controversy around megadoses, particularly for the common cold. What's the biochemical take on that?
Right, the Linus Pauling hypothesis. While large doses of vitamin C don't seem to prevent the common cold, there is some evidence they might slightly reduce the severity or duration of symptoms. This might be related to supporting immune cell function, particularly leukocytes, which use vitamin C.
But are there risks to taking huge doses?
The main concern is that a major metabolic breakdown product of vitamin C is oxalate. High vitamin C intake significantly increases urinary oxalate excretion. Since calcium oxalate is the main component of the most common type of kidney stones, there's a theoretical increased risk of stone formation in susceptible individuals taking megadoses long-term.
Good to know. Okay, let's plunge into the B-complex vitamins now, starting with the energy-releasing group. First up, B1, thiamine.
Thiamine. Its active coenzyme form is thiamine pyrophosphate, or TPP. TPP is absolutely central to carbohydrate metabolism and releasing energy from glucose. It specializes in oxidative decarboxylation reactions, removing a CO2 group from keto acids.
Where are the really crucial TPP-dependent steps in core metabolism, the ones that cause big problems if blocked?
There are two absolutely critical mitochondrial enzyme complexes that require TPP. First, the pyruvate dehydrogenase complex, PDH. This converts pyruvate, the end product of glycolysis, into acetyl-CoA, which then enters the TCA cycle. This links glycolysis to aerobic respiration.
Huge junction point. What's the second?
Second, the alpha-ketoglutarate dehydrogenase complex. This catalyzes a key energy-yielding step within the TCA cycle itself. TPP is also required for an enzyme called transcatolase in the pentose phosphate pathway, HMP shunt, which makes NADPH and precursor sugars.
So if TPP is deficient, you block pyruvate entry into the TCA cycle and you impair the cycle itself. That sounds like a massive energy crisis, especially for the brain, which relies so heavily on glucose.
It is a massive energy crisis, and that explains the classic deficiency disease, beriberi, historically common in populations relying on polished rice, which removes the thiamine-rich outer layers.
You mentioned beriberi comes in different forms.
Yes. In adults, it typically manifests as either dry beriberi or wet beriberi. Dry beriberi is primarily neurological. You see peripheral neuritis, nerve inflammation and damage, muscle weakness, maybe foot drop or wrist drop. This reflects the brain and nerves' inability to get energy from glucose efficiently.
And wet beriberi?
Wet beriberi involves the cardiovascular system. You get edema, swelling, tachycardia, fast heart rate, and potentially high-output cardiac failure because the heart is working inefficiently and trying to compensate.
Now, there's a specific, very high-yield clinical scenario involving thiamine deficiency, often linked to chronic alcoholism.
Yes, that's the Wernicke-Korsakoff syndrome, sometimes called cerebral beriberi. Chronic alcoholism severely impairs thiamine absorption and utilization, on top of often poor dietary intake.
What happens in the brain?
When TPP-dependent pathways are blocked in the brain, pyruvate and lactate build up, causing cellular damage. Clinically, Wernicke's encephalopathy presents acutely with confusion, ataxia, difficulty walking, and characteristic eye movement abnormalities, ophthalmoplegia and nystagmus. If untreated, it often progresses to Korsakoff psychosis, which involves profound memory loss, especially forming new memories, and apathy.
How is thiamine deficiency diagnosed biochemically?
The gold standard, especially for confirmation, is measuring the activity of the transcatolase enzyme in red blood cells, RBCs. Its activity will be significantly decreased in thiamine deficiency and will increase upon addition of TPP in the test tube.
Okay, very important clinically. Next energy releaser, B2, riboflavin.
Riboflavin. This forms the core of two crucial redox coenzymes, flavin adenine dinucleotide, FAD, and flavin mononucleotide, FMN.
Redox coenzymes, so they handle electron transfer.
Exactly. Their function is absolutely central to oxidation reduction reactions throughout all of metabolism. They participate by accepting or donating two hydrogen atoms, as FADH2 or FMNH2. The enzymes that use them are called flavoproteins, often recognizable by their yellow color.
Can you give us a key example of where FAD is essential, a really high-yield one?
A classic, absolutely essential example is succinate dehydrogenase. This enzyme catalyzes a key step in the TCA cycle, oxidizing succinate to fumarate. It's unique because it's also complex 2 of the electron transport chain. It directly links the TCA cycle to ATP synthesis via oxidative phosphorylation, and it requires FAD.
So deficiency would impair energy production. What does riboflavin deficiency look like clinically?
Deficiency is called araboflavinosis. It's often characterized by lesions of the skin and mucous membranes, places with rapid cell turnover. You typically see cheilosis, those painful fissures and cracks at the angles of the mouth, glossitis, a smooth, sore, often magenta or purplish-red tongue, and sometimes a scaly, greasy dermatitis, particularly around the nose and ears, nasolabial folds. Often occurs with other B vitamin deficiencies.
Right. Okay, next, B3, niacin, famous for preventing pellagra, the disease of the 3Ds.
That's right. Niacin is the precursor for two more vital redox coenzymes, nicotinamide adenine dinucleotide, NAD+, and nicotinamide adenine dinucleotide phosphate, NADP+.
How are NAD+ and NADP+ different in their roles?
Generally, NAD+ acts primarily in catabolic reactions, breaking things down. It accepts electrons, becoming NADH, during glycolysis, pyruvate oxidation, PCA cycle, fatty acid oxidation, and then carries those electrons to the electron transport chain to make ATP. NADP+, or rather its reduced form NADPH, is mainly involved in anabolic reactions building things up. NADPH is the major source of reducing power needed for biosynthetic pathways like fatty acid synthesis and steroid synthesis. It's also crucial for antioxidant defense via glutathione reductase.
Okay, catabolic NAD plus N, anabolic NADP plus M. Now, there's something unique about niacin synthesis, isn't there? We can actually make some.
Yes, this is critical. Niacin is unique among the B vitamins because the body can synthesize it, albeit inefficiently, from an essential amino acid, tryptophan.
How much tryptophan do you need?
The general rule of thumb is that about 60 milligrams of dietary tryptophan can be converted into roughly 1 milligram of niacin. This pathway requires other B vitamins like B6 and riboflavin, by the way.
So diets low in both niacin and tryptophan are double trouble, which leads us to pellagra.
Exactly. Pellagra is the classic niacin deficiency disease. It historically plagued populations whose staple diet was corn maize.
Why corn specifically?
2 reasons. First, the niacin present in corn is often chemically bound in a form called niacin, which isn't easily absorbed unless the corn is treated with alkali, like making tortillas with lime. Second, corn is naturally quite low in tryptophan, so you get hit from both sides.
And the symptoms, the famous 3Ds.
Yes, pellagra is characterized by the 3Ds. Dermatitis, a characteristic, often symmetrical rash on sun-exposed areas of the skin, looks like a sunburn that doesn't heal, sometimes called Casal's necklace around the neck. Diarrhea and dementia, which can range from irritability and confusion to severe psychosis. A 4th D, death, often followed if untreated.
Wow. Okay, now, niacin also has a completely different clinical use unrelated to its vitamin function, right, in high doses.
Absolutely. This is important pharmacologically.
Wow.
When given in very large doses, grams per day, way beyond vitamin requirements, niacin, specifically nicotinic acid, has a powerful effect on lipid metabolism.
What does it do?
It strongly inhibits lipolysis, the breakdown of fat in adipose tissue. By reducing the release of free fatty acids from fat stores, it decreases the liver supply of building blocks for making triglycerides. This, in turn, significantly lowers the liver synthesis and secretion of VLDL, very low-density lipoprotein.
And lower VLDL means?
Lower VLDL leads to lower LDL, bad cholesterol levels, and niacin also tends to raise HDL, good cholesterol. So, pharmacological doses of niacin are used therapeutically to treat certain types of hyperlipidemia, particularly type I hyperlipoproteinemia, to lower cardiovascular risk. Patients often experience flushing as a side effect, though.
Right, the niacin flush.
So,
Okay, next. B5, pantothenic acid. The name itself gives a clue about its availability.
yes, pantos is Greek for everywhere. Pantothenic acid is incredibly widespread in foods, making deficiency extremely rare in humans on normal diets.
Its coenzyme form is one of the absolute giants of metabolism.
It is indeed. Pantothenic acid is the core component of coenzyme A, often written as CoA or CoA-SH. Coenzyme is arguably the single most central molecule in all of intermediary metabolism. It acts as the universal carrier of activated acyl groups.
What's an acyl group and how does CoA carry it?
An acyl group is basically a fatty acid chain, like the 2-carbon-acetyl group from pyruvate. Coenzyme A has a reactive sulfhydryl group of SEH at its business end. This SSH group forms a high-energy thioester bond with the carboxyl group of an acyl group. The most famous example is acetyl-CoA.
Acetyl-CoA, the entry point for the TCA cycle.
Exactly, but CoA carries many other acyl groups too, involved in fatty acid synthesis, fatty acid oxidation, cholesterol synthesis, ketone body metabolism, you name it. Because acetyl-CoA sits at the crossroads of carbohydrate, fat, and protein metabolism, coenzyme A truly acts as the great metabolic integrator. Everything flows through CoA.
Given it's so fundamentally vital, why isn't deficiency a major clinical problem?
Simply because it's so ubiquitous in virtually all foods, meats, grains, legumes, vegetables. It's almost impossible to devise a diet deficient only in pantothenic acid. Deficiency has been induced experimentally, and in rare cases of severe malnutrition, it's been linked to a neurological condition called burning feet syndrome.
Burning feet syndrome.
Yes, characterized by numbness, tingling, and burning pain in the lower legs and feet, along with fatigue and weakness. But again, isolated deficiency is almost never seen clinically.
Got it. Okay, next. B6, pyridoxine. You called this the master regulator of amino acid metabolism.
I did, and it really is. Vitamin B6 actually exists as a group of related compounds, pyridoxine, pyridoxal, and pyridoxamine. But the active coenzyme form they all get converted to is pyridoxal phosphate, or PLP.
And PLP does what? Mainly with amino acids.
PLP is involved in a huge variety of enzymatic reactions, almost all of them involving transformations of amino acids. We can group its key functions into several major types of reactions.
Like what?
1. Transamination. This is absolutely crucial. PLP is required by aminotransferase enzymes like ALT and AST to swap amino groups between amino acids and keto acids. This links amino acid metabolism directly to carbohydrate metabolism, for example, making glucose from amino acid.
Okay, transamination. What else?
2. Decarboxylation. PLP is essential for removing the carboxyl group from amino acids to form biogenic amines. Many of these are vital neurotransmitters.
Examples.
For instance, PLP is needed to convert glutamate into GABA, gamma-aminobutyric acid, the main inhibitory neurotransmitter in the brain. It's needed to make serotonin from tryptophan, histamine from histidine, dopamine from dopa. All require PLP-dependent decarboxylases.
Wow, okay. Any other major roles?
3. Transsulfuration. PLP is involved in the pathway that synthesizes the amino acid cysteine from methionine. 4. Heme synthesis. The very first step in synthesizing for hemoglobin requires PLP. 5. Glycogenolysis. PLP is oddly also required for glycogen phosphorylase, the enzyme that breaks down glycogen.
Let's focus on that decarboxylation role in the neurotransmitters. What happens in B6 deficiency neurologically?
Because PLP is critical for synthesizing neurotransmitters, especially the inhibitory GABA, deficiency often manifests with neurological symptoms. In infants, severe deficiency can cause intractable convulsions or seizures due to the lack of GABAergic inhibition. In adults, you might see peripheral neuropathy, confusion, or depression.
And the heme synthesis link, does deficiency cause anemia?
Yes. Because PLP is needed for the initial step of heme synthesis, condensing succinyl-CoA with glycine, deficiency can lead to a specific type of anemia called sideroblastic anemia, which is typically microcytic and hypochromic, small, pale red cells, but with iron accumulation in the marrow.
Now, there's a really important drug interaction with B6, essential for clinicians to know.
Absolutely critical. The antituberculosis drug isoniazid, INH, is a structural antagonist of vitamin B6.
How does it antagonize B6?
INH reacts chemically with pyridoxal phosphate, PLP, forming an inactive complex. This effectively depletes the body's active B6 supply. Long-term INH treatment, if given without B6 supplementation, frequently causes peripheral neuropathy as a major side effect, directly due to the induced B6 deficiency.
So, standard practices.
Standard practice is to co-administer pyridoxine B6 supplements whenever prescribing isoniazid to prevent this neuropathy. Also, there's a diagnostic test for B6 status involving a tryptophan load. In B6 deficiency, tryptophan metabolism gets shunted down an alternative pathway, leading to increased excretion of xanthranic acid in the urine after a tryptophan challenge.
Good clinical pearl. Okay, last one in the energy-releasing group, B7 biotin, also known as the anti-egg white injury factor.
That's its historical claim to fame. Biotin's active coenzyme form is sometimes called biocytin, biotin linked to a lysine residue in the enzyme. Its unique and sole function is to act as a carrier of activated carbon dioxide, CO2, in a specific class of enzymatic reactions called carboxylation reactions.
Carboxylation, adding CO2. Where are these reactions important?
They are crucial at key metabolic crossroads, often involved in priming molecules for synthesis. Two absolutely vital examples are: 1. Pyruvate carboxylase. This enzyme converts pyruvate 3 carbons to oxaloacetate 4 carbons. Oxaloacetate is essential for replenishing TCA cycle intermediates and is the starting point for gluconeogenesis, making glucose. This reaction requires biotin and ATP.
Okay, pyruvate carboxylase for gluconeogenesis. What's the second key one?
2. Acetyl-CoA carboxylase, ACC. This enzyme catalyzes the first committed step in fatty acid synthesis. It carboxylates acetyl-CoA to form malonyl-CoA. This reaction also requires biotin and ATP. So biotin is essential for both making glucose and making fat.
Right. Now, about that egg white thing, how can raw eggs cause a deficiency?
Raw egg white contains a glycoprotein called avidin. Avidin has an incredibly high binding affinity for biotin, one of the strongest non-copulsaric interactions known in biology. It binds biotin extremely tightly in the digestive tract.
And prevents absorption.
Exactly. If you consume large amounts of raw egg white over a prolonged period, the avidin effectively sequesters all the dietary biotin, preventing its absorption and leading to deficiency symptoms, dermatitis, hair loss, neurological issues. Cooking egg whites denatures the avidin, destroying its ability to bind biotin. So cooked eggs are perfectly fine. This interaction was actually key to discovering biotin's essential role.
Fascinating. Okay, we've covered the energy releasers. Now let's move to the final B-complex subgroup, the hematopoietic group. That's folic acid, B9, and cobalamin, B12. You said these two are intricately linked clinically. Let's start with folate.
Right, folic acid, or folacin, its vital role is in one-carbon metabolism. The active coenzyme form is tetrahydrofolate, THF.
What does THF do?
THF acts as a carrier for various single carbon units, things like methyl groups, CH3, formal groups, CHO, methylene groups, CH2. It picks up these one-carbon fragments from donor molecules and transfers them to acceptor molecules in a wide range of biosynthetic reactions.
Where are these one-carbon transfers absolutely critical?
They're essential for the synthesis of several key building blocks. Most importantly, 1. Purines. THF derivatives provide 2 carbon atoms for building the purine ring structure, adenine and guanine, which are needed for both DNA and RNA. 2. Thymidylate, DTMP. THF is required for converting DUMP into thymidylate, DTMP. Thymine is the unique base found only in DNA, so folate is directly required for DNA synthesis.
DNA synthesis. That immediately tells you where deficiency symptoms will show up first.
Exactly. In any tissues with rapid cell division where DNA synthesis is high, the prime example is the bone marrow, where blood cells are constantly being produced.
What happens in the bone marrow if folate is deficient?
DNA synthesis is impaired, but RNA and protein synthesis continue somewhat normally. This leads to an imbalance in cell growth and division. Cells grow large but cannot divide properly. In the red blood cell lineage, this results in the production of abnormally large, immature red blood cells called megaloblasts. When these enter the circulation, you get macrocytic or megaloblastic anemia.
Is folate deficiency common?
Yes, it's considered one of the most common vitamin deficiencies worldwide, particularly in pregnant women and individuals with poor diets or malabsorption.
And the pregnancy link is critical, right? Neural tube defects.
Absolutely critical. Folate deficiency during early pregnancy dramatically increases the risk of severe neural tube defects, NTDs, in the developing fetus, such as spina bifida and anencephaly. This is why folic acid supplementation is strongly recommended before and during pregnancy.
We also need to connect folate to cardiovascular risk through homocysteine. How does that work?
Right. This is a key concept. Both folate, specifically N5-methyl-THF, and vitamin B12 are required cofactors for the enzyme methionine synthase. This enzyme converts the amino acid homocysteine back into methionine.
Okay.
If either folate or B12 is deficient, this reaction slows down and homocysteine levels build up in the blood. This condition is called hyperhomocysteinemia. Elevated plasma homocysteine is now recognized as an independent risk factor for cardiovascular disease, atherosclerosis, heart attacks, strokes.
So folate helps keep homocysteine levels down. Good to know. And finally, antagonists, drugs that interfere with folate.
Yes, very important clinically. 1. Methotrexate. This is a major anti-cancer drug. It's a structural analog of folate and powerfully inhibits the enzyme dihydrofolate reductase, DHFR. DHFR is needed to regenerate active THF from its oxidized form, BHF. By blocking THF regeneration, methotrexate shuts down DNA synthesis, preferentially killing rapidly dividing cancer cells, but also affecting normal dividing cells, causing side effects.
And the other type of antagonist?
Here, sulfonamides. These are antibiotics. Bacteria, unlike humans, need to synthesize their own folic acid from a precursor molecule called PABA, paraminobenzoic acid. Sulfonamides are structural analogs of PABA and block the bacterial enzyme that incorporates PABA into folic acid. Humans don't use PABA, so these drugs selectively inhibit bacterial growth.
Clever. Okay, our last individual vitamin, B12 cobalamin. You flagged this one as unique in several ways.
It is quite unique. First, B12 is the only vitamin we know of that is synthesized exclusively by microorganisms, bacteria and archaea. Plants and animals cannot make it at all. We rely entirely on dietary intake from animal products, meat, fish, dairy, eggs, or fortified foods or supplements.
What else is unique?
It's the largest and most complex vitamin structure. And significantly, it's the only vitamin that contains a trace metal ion, specifically cobalt, coordinated within a corin ring structure, similar to the porphyrin ring in heme, hence the name cobalamin.
And its absorption is also highly specialized, requiring that extra factor.
Yes, B12 absorption is a complex process absolutely dependent on intrinsic factor, IF. IF is a glycoprotein secreted by the parietal cells of the stomach lining, the same cells that secrete acid.
What does intrinsic factor do?
It binds tightly to dietary B12 in the stomach, protecting it from digestion. The B12-IF complex then travels down to the terminal ileum, the last part of the small intestine, where specific receptors recognize and bind the complex, allowing B12 to be absorbed into the bloodstream. Without IF, B12 absorption is virtually zero.
And unlike other water-soluble vitamins, B12 is stored.
Yes, that's the other major exception. The body is very efficient at conserving B12. Significant amounts are stored, mainly in the liver. These stores can last for a remarkably long time, typically 4 to 6 years before deficiency symptoms appear, even if intake stops completely.
Wow, years. Okay, functions. In mammals like us, B12 is only known to be a required coenzyme for two enzymatic reactions. What are they?
Only two, but they are critical. One, methionine synthase. We just mentioned this one. It requires methylcobalamin, a form of B12, as a cofactor to transfer a methyl group from N5-methyl-THF to homocysteine, regenerating methionine and, crucially, freeing up active THF. This directly links B12 and folate metabolism.
Okay, methionine synthesis, what's the second reaction?
2. Methylmalonyl-CoA. This enzyme requires deoxydenadosocobalamin, another form of B12. It catalyzes the isomerization rearrangement of L-methylmalonyl-CoA into succinyl-CoA.
Why is that reaction important?
Methylmalonyl-CoA is produced during the breakdown of odd-chain fatty acids and certain amino acids like valine, isoleucine, threonine. Converting it to succinyl-CoA allows these breakdown products to enter the TCA cycle for energy. If this step is blocked by B12 deficiency, methylmalonyl-CoA accumulates.
Okay, two key reactions. Now, deficiency. The classic disease is pernicious anemia. Is that usually due to a lack of B12 in the diet?
Usually not, especially in Villet countries. Pernicious anemia is classically defined as megaloblastic anemia resulting from lack of intrinsic factor, not dietary B12 deficiency.
Why would someone lack intrinsic factor?
Most commonly, it's due to an autoimmune attack against the gastric parietal cells that produce IF, or against IF itself, autoimmune gastritis. This leads to failure of B12 absorption, regardless of intake. Other causes can include surgical removal of the stomach, gastrectomy, or parts of the ileum where absorption occurs.
So pernicious anemia causes the same megaloblastic anemia as folate deficiency.
Yes, because the block methionine synthase reaction leads to that functional folate deficiency we'll discuss. But critically, B12 deficiency also causes neurological symptoms, which folate deficiency typically does not.
What kind of neurological symptoms, and why do they happen in B12 deficiency?
Symptoms can include numbness and tingling in the hands and feet, paresthesia, difficulty walking, ataxia, memory loss, confusion, depression, and eventually irreversible nerve damage, subacute combined degeneration of the spinal cord. The neurological damage is thought to be primarily due to the accumulation of methylmalonyl-CoA because the mutase reaction is blocked. High levels of methylmalonyl-CoA interfere with normal myelin synthesis and maintenance, leading to demyelination.
Right. That second B12 reaction is key for the neurosymptoms. Okay, this brings us to the absolute crux of the clinical interaction, the folate trap, or methyl trap hypothesis. Can you explain this? It's complex but essential.
It is complex but critical for understanding diagnosis and treatment. Remember the methionine synthase reaction. It takes the methyl group from N5-methyl-THF and gives it to homocysteine, using B12, regenerating active THF.
Got it.
Now, N5-methyl-THF is formed essentially irreversibly from other THF forms. The only significant way for the body to get that methyl group off N5-methyl-THF and regenerate the usable THF pool needed for purine and thymidylate synthesis is via the B12-dependent methionine synthase reaction.
So if B12 is deficient?
If B12 is deficient, the methionine synthase reaction grinds to a halt. As other THF forms get converted to N5-methyl-THF, carrying 1 carbon units for metabolism, they get stuck there. They can't be demethylated back into the active THF pool.
Yeah.
So almost all the body's folate gets effectively trapped in the unusable N5-methyl-THF form.
So even if total folate levels in the tissue might be normal or even high, there's no active THF available for DNA synthesis.
Precisely. This creates a functional folate deficiency at the level of DNA synthesis, even with adequate folate stores. And that's why B12 deficiency causes the same megaloblastic anemia as primary folate deficiency. The underlying biochemical block for anemia is the lack of active THF derivatives.
This has massive clinical implications for treatment, doesn't it? It sounds like a diagnostic pitfall.
It is a huge potential pitfall. Imagine a patient presents with megaloblastic anemia. You run tests. If you were to treat them with high doses of folic acid alone, what would happen?
Well, high doses of folate might provide enough substrate to sort of bypass the trap or push other pathways, maybe correcting the anemia.
Exactly. Giving large amounts of folic acid can overcome the functional block and provide enough THF derivatives through other minor pathways to allow DNA synthesis to resume in the bone marrow. So the anemia might improve or even resolve, the blood picture looks better, but you have done absolutely nothing to fix the underlying vitamin B12 deficiency. The methionine synthase reaction is still blocked. More importantly, the methylmalonyl-CoA mutase reaction is still blocked.
So the neurological damage continues.
The devastating, often irreversible neurological damage caused by the accumulating methylmalonyl-CoA continues unchecked, even while the anemia is masked by the folate treatment. This is incredibly dangerous. Treating megaloblastic anemia with folate without ruling out B12 deficiency can lead to severe permanent neurological disability.
So the absolute rule is?
The rule is, never treat megaloblastic anemia with folic acid alone until you have definitively excluded vitamin B12 deficiency as the underlying cause. Standard practice is often combined B12 and folate therapy is unsure, but ensuring adequate B12 is paramount, especially if neurological symptoms are present. B12 status must be assessed.
Okay, let's try to consolidate some of this vast knowledge into actionable clinical principles. We've covered a huge amount of ground, from basic chemistry to complex pathways and deficiency states. What are the absolute core takeaways for management?
Well, building on that last point, remember the key treatment strategies. For pernicious anemia, because the root cause is lack of IF causing malabsorption, oral B12 is usually ineffective. Treatment requires intramuscular or subcutaneous B12 injections to bypass the gut entirely.
And often combined with folate?
Often, yes, especially initially, to address the functional folate deficiency quickly. But the B12 is the non-negotiable part to prevent or manage the neurological decline.
What about using vitamins prophylactically to prevent deficiency?
Key examples include B6 pyridoxine supplementation must be given concurrently with the anti-TB drug isoniazid, INH, to prevent that drug-induced peripheral neuropathy. It's standard care.
And in pregnancy?
High-dose folic acid supplementation is absolutely vital before conception and during early pregnancy to drastically reduce the risk of neural tube defects. This is a major public health intervention.
And don't forget the non-vitamin use of niacin.
Right. Pharmacological doses of niacin remain a tool, often in combination therapy, for managing certain types of hyperlipidemia, specifically for lowering VLDL and LDL and raising HDL due to its powerful inhibition of lipolysis.
We briefly touched on a few compounds that act a bit like vitamins but aren't strictly essential, sometimes called vitamin-like compounds. Can we revisit lipoic acid quickly?
Sure. Lipoic acid or alpha-lipoic acid is fascinating. It's unique because it's soluble in both water and fat, earning it the nickname universal antioxidant.
Does it have a coenzyme role too?
Yes, it acts as a coenzyme specifically involved in those same oxidative decarboxylation reactions as TPP. It's part of the pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase complexes involved in the acyl group transfer. But beyond that, it's gaining a lot of interest for its potent free radical scavenging abilities throughout the body, including the brain.
So therapeutic potential.
High doses are being actively researched for potential benefits in conditions involving oxidative stress, like diabetic neuropathy, Alzheimer's disease, stroke recovery, and for stimulating the body's own production of glutathione, another key antioxidant. Still largely investigational, but interesting.
Are there any others that blur the line between vitamin and metabolite?
Two others often mentioned are choline and anisitol. Both can be synthesized by the body, so they aren't strictly vitamins. But sometimes dietary intake becomes important under high physiological demand.
What are their roles?
Choline is a crucial component of phospholipids like phosphatidylcholine or lecithin in cell membranes. It's also the precursor for the neurotransmitter acetylcholine. And it acts as a lipotropic factor, helping to prevent fat accumulation in the liver. Inositol is also a key component of cell membrane phospholipids, phosphatidylinositol, and is fundamental to intracellular signaling pathways as a precursor for second messengers like IP3 and DAG, which are triggered by many hormones.
Right. Synthesizable, but still important. Okay, time for a quick quiz to help reinforce some key connections. Let's link the deficiency back to the patient presentation. Question 1. Which B-complex vitamin deficiency, often seen in chronic alcoholics, typically presents with a triad of confusion, difficulty walking, ataxia, and characteristic abnormal eye movements? And what core metabolic process is failing?
That classic triad points strongly towards thiamine, B1 deficiency, manifesting as Wernicke-Korsakoff syndrome. The core process failing is TPP-dependent oxidative decarboxylation, primarily of pyruvate and alpha-ketoglutarate, leading to a severe energy deficit in the brain and accumulation of lactate.
Perfect. Second question. Can you recall the name of the single enzyme that humans lack, preventing us from synthesizing vitamin C and making it an essential dietary nutrient?
That would be L-galonolactone oxidase. Its absence in our evolutionary lineage means we absolutely must get vitamin C from our diet. Fantastic job. Seriously, you've just worked through and hopefully mastered some really key high-yield concepts in biochemistry. The goal now is to be able to connect these molecular players, these vitamins and coenzymes, directly to patient health and disease. Understanding the deficiency isn't random. It happens because fundamental metabolic pathways literally break down.
Yeah, that's the core message. And for our final provocative thought today, let's circle back to that B12 and folate relationship, the folate trap. We saw how the exact same clinical sign, megaloblastic anemia, can stem from two entirely different vitamin deficiencies. The folate trap hypothesis is a powerful reminder that treating just the symptom, the anemia, with the wrong tool, folate alone, can actually mask the deeper, more dangerous problem, the underlying B12 deficiency, and the risk of permanent neurological damage that folate won't fix. It really underscores that context, codependence, and understanding the whole pathway are absolutely everything in biochemistry and medicine.